Our next competitor is Marcus Giacci. Marcus is in the second year of his PhD in the School of Animal Biology and is supervised by Associate Professor Lindy Fitzgerald and Associate Professor Nathan Hart. His thesis title is Fate Mapping and Protecting Oligodendrocyte Progenitor Cells in Areas of Secondary Degeneration Following Neurotrauma. His DMT title is Investigating Damage to Myelin Stem Cells Following Neurotrauma. Marcus Giacci.
Now, most of you have heard about neurons, the cells in your nervous system that transport information around your body. But have you heard about myelin? Myelin is a structure that tightly wraps around the length of your neuron, and this structure is incredibly important because it allows your neurons to speak to each other effectively. Without this myelin structure, your ability to see, walk, or think may be affected. In a healthy nervous system, the myelin tightly wraps around the neurons, like the picture in the top left. However, there are certain conditions which may cause this structure to be compromised. One such condition is neurotrauma, that is brain and spinal cord injury. Now, neurotrauma can be broken into two main phases, the initial phase and the secondary phase. The initial phase may be brought about by an impact to the head, as pictured in the center. It is the secondary phase, however, that causes a majority of the devastating effects of neurotrauma, and this can last for years after the initial impact. The secondary phase is caused by masses of oxidants spreading from the initial injury site to the adjacent uninjured areas, damaging DNA and destroying structural proteins like those found in neurons and myelin. Now, a key feature of this secondary phase is that the tight myelin wrapping around your neurons becomes loose, like the image in the top left for you guys. Now, one of the main reasons that this is thought to occur is because the myelin stem cells that normally replace and repair damaged myelin in a normal healthy nervous system die following neurotrauma due to the exposure to these oxidants. So I wanted to know if the masses of oxidants that are present following neurotrauma are damaging these myelin stem cells and causing their numbers to decrease. To study this, I induced neurotrauma by partially cutting a nerve in a rat. I then investigated the areas affected by the secondary phase of injury around this first cut. The blue histogram, looking at the DNA damage in these myelin stem cells, sees a significant increase as early as 3 days after the initial cut. I also counted the numbers of these cells and found that their numbers significantly decreased 7 days after injury. This is the first time that this oxidant damage has been assessed in these cells following neurotrauma. And now that I've identified a source of damage to these cells, I can look at ways to treat them. I'm currently investigating antioxidant therapies to reduce the damage done to these myelin stem cells by oxidants. This could preserve the numbers of these myelin stem cells and allow them to go on and replace damaged myelin after neurotrauma. This could be the difference between improper and proper functioning of neurons and could potentially restore the ability to see, walk, or think for people who have suffered neurotrauma. Thank you.